Okay, so this is a revision video on digestion and absorption of proteins. So there are two revision tasks. The first one is to describe endo, exo and dipeptidases, how they work and their product. And then secondly, you should be able to describe protein digestion and absorption, and you should be referring to specific organs, enzymes and transport processes involved. So pause and test yourself on those, and then we'll go through answers. Okay, so question 1, what you should have said for endopeptidases is this. So it's the central peptide bonds that are hydrolyzed by endopeptidases. So just a diagram to show you. We've got a polypeptide or protein, amino acids and peptide bonds. And so those arrows indicate where an endopeptidase would work. Just those central peptide bonds. What would be the product of that? Smaller peptides. And so your answer for exopeptidases should be this. So exopeptidases work on the end or terminal peptide bonds, as they're known. So in this diagram, they're going to be working on those end peptide bonds in those smaller peptides. And so what's produced? Amino acids, but also some dipeptides as well. And so finally, your answer for dipeptidases should be this. And remember, those dipeptidases are actually membrane bound. We'll come to that in a moment. So obviously, dipeptidases work on the peptide bonds between dipeptides, and so the final products you have are the monomer of polypeptides or proteins, amino acids. Okay, so let's go through part 2. So the first organ you should have mentioned is the stomach. And in the stomach, it's the endopeptidase pepsin that, remember, works at an optimum pH of around 2 because of the hydrochloric acid that's secreted into the stomach. And that hydrolyzes polypeptides into smaller peptides. We've just covered that they work on the central peptide bonds. So the next organ you should mention is the pancreas. So the pancreas makes endopeptidases and exopeptidases. They're secreted in pancreatic juice, and the pancreatic juice makes its way into the duodenum. That's the next organ you should have mentioned, or part of the small intestine. And that's where the endopeptidases and exopeptidases hydrolyze polypeptides into smaller peptides. And exopeptidases work on those terminal peptide bonds to hydrolyze those terminal peptide bonds to produce dipeptides and amino acids. So then contractions of the small intestine move those products along, those amino acids and dipeptides, and they reach the ileum. And that's where absorption occurs. So let's focus in on that and what you should have described for that. Of course, that's the small intestine. And so at the ileum, it's the membrane-bound dipeptidases that then produce amino acids by hydrolyzing the peptide bonds in dipeptides. Right, now let's look in more detail at the absorption of amino acids. So we've got the epithelial cells in the ileum with the microvilli that increase the surface area for absorption. In our picture, we've got a dipeptide and the dipeptidase remember it's membrane bound, hydrolyzes the peptide bond to produce amino acids. The important thing is there are sodium ions in the lumen of the ileum, and obviously they're the amino acids that have been produced by digestion. So this process is the same as for absorption of monosaccharides. We looked at a specific example of glucose. So now we're just going to go through again for amino acids. So step 1, there is a higher concentration of sodium ions in the blood than in the cytoplasm of the epithelial cell. Sodium ions are actively transported out through a sodium-potassium pump into the blood. Potassium moves in. Now we have a lower concentration of sodium ions in the cell than in the lumen of the ileum. So this is step 1. So now sodium ions from the lumen of the ileum can diffuse down their concentration gradient through a co-transport protein. This is step 2. And that allows amino acids to move in against their concentration gradient, along with sodium ions in this sodium amino acid co-transporter. So we get sodium ions moving in by facilitated diffusion, well, co-transport really, but they're diffusing down, and amino acids moving against their concentration gradient using this co-transporter. Step 2. So finally, step 3, we now have a high concentration of amino acids compared with the blood. So a high concentration of amino acids in the cytoplasm of the epithelial cell compared with the blood. So these can diffuse down their concentration gradient into the blood through a carrier protein by facilitated diffusion. That's how amino acids end up in the blood. That's step 3, described there. Okay, so that is the process of digestion of proteins and absorption of the amino acids from that digestion. So if you didn't get that, I suggest you go back, repeat, test yourself, and keep testing yourself until you're getting it right consistently.